This is the second example, which is a two-stage design with dose selection. So let me take you back to 2007 when I was at Napo Pharmaceuticals. We were designing a trial. We were given the go-ahead by the FDA to go into phase 3, and we had a real problem. We had the designation of one trial to get approval, but the company had planned on 2 doses, and looking at the data, it wasn't so certain. So the idea came up of planning a 3rd dose, and you can imagine the cost of doing that in terms of powering that, and then you have multiplicity and the like. So next slide. We looked at different options, and I'm sure you're familiar with this. Number 1 was to run a single 4-arm trial, which would be 4 dose, which would be actually 3 dose groups versus placebo. The second would be to go back to phase 2 and run a phase 2 trial and then move into an independent phase 3. And the third option we took was the seamless phase 2, which would be to start with 4 arms, select the best dose at an interim analysis, and then continue with 2 arms, 1 active dose and 1 placebo, which is what we chose. Next slide. So let's look at the sample size. The most conventional, which would have been to run a 4-arm trial, would have been with a Bonferroni adjustment, 450 patients, something the company couldn't afford. The second was to run 2 separate trials, which would have actually been a lower sample size in aggregate of 416 patients. We would have added a year to a year and a half onto the timeline. And based on the fact that we were a public company at that time and had already announced to the street that we were doing a phase 3 trial, not something that we really wanted to consider. And the last was a 2-stage design where we used the Posh-Bauer estimate. It basically allows you to pool p-values between the first stage of your study where you have 4 arms and the second stage of your study when you have 2 arms. And that's an accepted statistical methodology by FDA. And with that, we were able to get down to 350 patients, which is the option we chose. Next slide. So this shows you this is a schematic. We started with 4 doses. We went to an interim analysis. The principles that Adam just discussed about the interim analysis, I'm going to repeat. It has to be vigorous. You have to have a very clear plan in place with the FDA. You have to show them you're going to have complete blinding and that you're not going to be unblinded to the data. So we actually had a dose selection committee. You might call it a DMC, although they didn't adjudicate safety with that. But they selected a dose, and we actually gave them very rigid rules for picking the dose. And the final rule that they actually resorted to was that if all the doses looked similar, and we defined similarity as within 2% of each other, pick the lowest dose. And that's exactly what they did. Next slide. So the outcome was that we did develop a SAP that went to the agency ahead of time. We convinced the agency, as Adam mentioned, that we were controlling the type 1 error, which is what we did with the statistical analysis we planned. We had a SAP approved by the FDA through a special protocol assessment. The trial completed in 2010 and was actually the lowest of the 3 active arms that was selected by the Interim Analysis Committee. The p-value was less than 0.01, and the drug went on to approval at the end of last year. Next slide. So what are the take-home messages? The first is that you have to convince the agency, whether it's the 2-stage design or whether it's the sample size re-estimation design, that you've controlled the type 1 error. You have to convince them that you have rigorous control of your blinding and your interim analysis, and there must be a pre-specified charter. You have to have an independent statistical center in order to oversee that process. That's the key in both of these designs, and that you have to have rigorous procedures in place in order to assure that you have rigorous preservation of blinding.